Okay, here we are for the cell division video, otherwise known as the process of mitosis. Because cells are the smallest unit of life, cells must be able to do all of the things that living things can do. If you think back to the beginning of the school year, we talked about 8 characteristics that all living things share. All living things must be able to get and use energy. All living things must be able to adapt to their surroundings. Okay, if you think back, we can come up with all 8 of those, but most importantly, all living things must be able to reproduce. Mitosis, or cell division, is the cell's way of reproducing. Okay, mitosis is the description of this process that produces 2 identical cells as a result of cell reproduction. Before we get into the actual process of mitosis, it is necessary to review the cell cycle or the cell life cycle that we did in stage 1. After we have a division of a cell right here, the cell life cycle continues in this clockwise manner. Excuse me. So after the division here in G1, the cell is growing, it is developing. It is performing its job, but it is also growing in size and developing as a cell. Once it has reached the maximum size, it needs to prepare to divide. Part of that preparation is going to include reproducing the DNA, or doubling its DNA for division. Notice here, at the end of G1, we have a single piece of chromosomal material, or in this case, 2 single pieces. This is one chromatid. Notice after the S phase, the synthesis phase, where the DNA is doubled, we have a full chromosome, this X-like structure right here. Here, singular. Oops, that was a bad arrow. Here, after the DNA has synthesized or doubled, we have one chromosome, or 2 sister chromatids put together in this X-like shape. Okay, here singular, here doubled. Now we are ready for reproduction. Okay, so during this phase here, interphase, the cell is still doing its normal job. Muscle cells are flexing, hair cells are being hair, skin cells are covering the body. Okay, there's only a small part of the cell cycle that is actually cell division. Okay, this right here would be the cell division section, and that is what we're going to cover in this video. This is mitosis, this is cell division. Sometimes this is referred to as simply the M phase. Notice that when we focus on this sliver of the cell cycle, there are many processes that happen during the process of cell division. We're going to try and break this down in a way that makes them as understandable as possible. You can relate the process of cell division to the process of moving from one house to another. If you're going to move from one house to another, you'd have to pack up and organize yourself, which is what's going to happen through these phases. Okay, from there, you will literally have to move from one place to another, which is happening the rest of the way. And you will see this as we break down these stages step by step. Here we begin the first phase of mitosis with prophase. Pro here means before. Like when we talked about prokaryotic organisms, we know that they existed before the nucleus. So here we have prophase, before the division. What are the details happening during prophase? The first thing that you can notice is that this nuclear membrane is beginning to dissolve. It's breaking apart. If we relate this to our moving analogy, this would be taking the legs off of the tables so they'll fit out the door. This would be taking the hinges off the door so the couch fits out. You have to do things like that in order to make the move simpler. We also notice, and this is kind of done already in the picture, but our DNA has condensed. The chromatin has condensed into chromatids, and those are connected into what we call chromosomes. This is nature's way of taking that DNA and packing it with the little styrofoam peanuts. Okay, like you would take your glassware, your dishes, and wrap them or stuff them with peanuts so they don't break. Same thing is happening with the DNA right here. You can also notice that the centrioles, you can't really see it in this picture, but what they're doing is they're moving to opposite sides of the cell. Okay, and this will help with the actual division part, as these spindles that are coming off of the centriole will help pull the chromosomes apart and make two different cells. Toward the end of prophase, or what we call late prophase, we see that the nuclear membrane is completely dissolved. The cell wants to connect the centrioles to the centromeres of the chromosomes. Okay, if we look at a chromosome here, this entire structure is a chromosome. Each chromosome is made of two chromatids. Okay, so you have one chromatid and another chromatid. Where they are connected in the middle, or the center of the X shape, is what we call the centromere. Okay, so the cell wants to connect these centrioles up here to the center of that chromosome, or the center that we know of called the centromere. Centromere. Okay. This connection here forms a spindle, and like I previously said on the last slide, this will actually pull those chromosomes apart when we want two different cells. Okay, this is the cell packing up, and this is the cell organizing for it to move. This packing up and organizing will finish in the beginning of metaphase. Okay, here we have metaphase, which is basically the middle process. If we were to pack up and move from one home to another, we'd have the organizing and packing phase, and then we would have moving to the new house and unpacking there. This phase would be in the middle of those two. Essentially, you could relate this to having your entire house packed up, all your boxes labeled, and then setting those boxes into the moving truck, ready for that physical part of the move. Okay, we have everything nice and tightly packed in the center of the cell here. You can notice that the chromosomes are all lined up in the center of the cell. The spindle fibers are attached to the centromeres of those chromosomes. The centrioles are on opposite sides of the cells, and those are ready to push and pull the chromosomes apart, so one half of that chromosome can go to each new cell. From there, we get to go to anaphase. Okay, anaphase you can refer to as the action phase. This is where we actually get some of that separation, we get some of that division. Okay, relate it back to our move. Okay, we had the boxes all loaded up, we had the boxes labeled, they were in the moving truck. This would be taking that truck and physically driving it to the new place. Okay, so now what's happening here is during mitosis, each one of these chromosomes is going to split into two. All right, half of that chromosome is going to go to one cell, and half is going to go to the other. All right, so you can notice here, you had one chromatid split from its sister chromatid, one of that with all of the DNA for one complete cell will end up in this cell, and the other half will end up in this bottom daughter cell. Okay, so here we are. This would be at the end of our move when we want to begin unpacking our boxes. Telophase is the portion of cell division where we begin unpacking. All right, you can kind of notice in this picture that the DNA material has begun to unwind. They're not as densely packed anymore. They're beginning to unwind and become less dense. You can also see here that the nuclear membrane is returning. It's starting to form back again, keep that DNA inside of the nuclear membrane. We also get what is called a cleavage furrow, where that cell membrane begins to pinch the two new cells apart. Okay, so that is basically what happens in telophase. The end result, we get two cells with the same materials inside of them. They are exact copies of each other. And the final process in cell division is what we refer to as cytokinesis. Okay, in telophase, we had the nuclear membrane forming, the DNA is unwinding, and now all we have to do is physically separate the two cells apart from each other. Okay, so cytokinesis is dividing the cytoplasm. We are taking this cleavage furrow that was formed in telophase, and we keep pinching that cell all the way down, so we have two separate cells at the end of cytokinesis. Usually this is happening at the same time as telophase, because as you can see, they're very closely related. The timing is almost identical. What we haven't talked about is the difference between plant and animal cells. Okay, so what you've seen so far is referring to animal cells, where the cell membrane pinches off into two different cells. When we talk about animal cells, really the only difference is this cytokinesis part, where the two cells must separate themselves. So if we look at this picture, where the difference comes in is that the cell wall of a plant is rigid. Okay, we've talked about that. It helps hold the cell or the plants upright, as it has to be rigid in order for them to stand up. Because that is rigid, that membrane cannot pinch itself off to form two new cells. So what happens in plants is, instead of pinching off, a cell plate forms and kind of grows between the two new nuclei. This allows it to separate the contents of the cell. It separates the cytoplasm. It separates our two new daughter cells that have been formed in the plant. So you can kind of see one forming here in this microscope picture, separating the newly formed materials. Okay, so in the end, the results we get from mitosis. In unicellular plants and animals, it results in new offspring by asexual reproduction. Okay, think of bacteria. Bacteria undergo asexual reproduction where they are basically reproducing by making exact copies of themselves. In multicellular organisms, it helps us, for example, grow and repair the organism. As you grow as an embryo, you are dividing cells, you're developing that way. If you get a cut on your arm, our skin cells divide and repair that skin. Okay, and it's important for a few reasons. Obviously more than a few, but the main ones. The two new cells are exact duplicates of the mother cell. One mother cell divides and creates two new daughter cells exactly the same. This ensures that the new cells will be able to carry on the same functions as the mother cell. If you have a skin cell dividing, you want the two daughter cells to be able to do the same function that the skin does. Protects you water barrier, that sort of things. Okay, so now I'm going to end this video with an opportunity for you to practice identifying the stages. Okay, so remember we had interphase of the cell. After it divides, it grows and develops and copies its DNA. We have prophase, we have metaphase, we have anaphase, and then we have telophase and cytokinesis. So I'm going to show you this picture right here, and I would suggest that you pause the video right now and maybe try to jot down what the number ones would be, what the number twos would be, what the number threes would be, and fours and fives. Okay, so you can see everything that has a number 1 by it looks fairly similar. Okay, I would like you to try to tell me what phase that is. The number 2s, there's only one phase, it looks like, on this slide. Oh, here's one up here. Okay, tell me what that phase is. So take a second, pause it, write all these down, and I will flip through the answers here for you in just a second. So the number 1s, we can see that these are in prophase. That DNA has condensed. We can kind of see it's a lot denser than maybe this cell right here. Number 2s, we have metaphase. Especially in this one right here, we can see that the chromosomes have lined up in the center of the cell. Number 3, we have anaphase. This is a good picture down here where the chromosomes are being pulled to different sides of the cell. Number 4, we have telophase. All right, we can kind of see that we have 2 separate nuclei forming. And then number 5, we have interphase. This is the only number 5 I see. We have a cell during its growth and development stage. This cell is just performing its role. Okay, we do not see dense DNA in the center. So we can say that is an interphase, just performing as whatever cell this is.